Hi everyone, welcome back to my channel. In continuation in the previous video, today we'll learn about steps in transcription. The process of transcription in the case of prokaryotes and eukaryotes is quite different. That's why I'm going to discuss it in two different videos. In this video, I'm going to just talk about the process of transcription in prokaryote. The process of transcription in the case of prokaryote can be studied under mainly three headings, that is initiation, elongation and termination. We'll discuss these three steps one by one. First one is initiation. As I already told in the previous video, in the case of prokaryote, the synthesis of all three types of RNA, that is mRNA, tRNA and rRNA, is done with the help of only one enzyme known as RNA polymerase. It is also called as DNA dependent RNA polymerase. So RNA polymerase has the ability to produce all three types of RNA, and these three types of RNA are very essential for the translation process or during protein synthesis process. So these three types of RNA need to be produced during transcription with the help of enzyme known as RNA polymerase, and synthesis of this RNA begins in the first step, that is initiation step. In the initiation step, the RNA polymerase enzyme go and bind to the promoter site. Promoter site is nothing but the small sequence of DNA in the transcription unit. Transcription unit means what? It's a stretch of DNA or small sequence of DNA that need to be transcribed into RNA. So in this transcription unit, the promoter region is present, or promoter region is a small sequence of DNA that is present in the transcription unit to which this RNA polymerase go and bind. Binding of this RNA polymerase to the promoter region is done with the help of a sigma factor. Sigma factor is a small part of RNA polymerase enzyme itself. This sigma factor will recognize this promoter site and it helps the binding of RNA polymerase to the promoter site of the transcription unit. Once this RNA polymerase bound to the promoter site, it catalyzes the unwinding of the DNA double helix. So the DNA double helix at this part, now it starts to unwind or separate, which provide free DNA template strand for the addition of a ribonucleotide. So once the free DNA template strand is formed, RNA polymerase enzyme catalyzes the addition of a ribonucleotide. Now, the ribonucleotides are added to this free DNA template strand based on the complementarity. So, based on the complementarity, new ribonucleotide strands are added to this free DNA template strand, and it is catalyzed by RNA polymerase enzyme. This RNA polymerase enzyme or holoenzyme will be attached to the promoter site until 8 or 9 bond is formed, or until 8 or 9 ribonucleotides are added to this template strand. This holoenzyme or RNA polymerase enzyme will be attached to this promoter site. Once 8 or 9 bond is formed, this sigma factor will be released. Then only core enzyme part, which has 2 alpha subunit and beta dash subunit, which is a core enzyme part of the RNA polymerase, now will proceed with the second step, that is elongation step. In the elongation step, there is an elongation of RNA strand which is catalyzed by RNA polymerase core enzyme after the release of a sigma factor. In the elongation step, this RNA polymerase will move along the DNA template strand, during which for each exposed base of the DNA template strand, it will add a ribonucleotide based on the complementarity. So RNA polymerase will add a ribonucleotide to the 3' OH end of the previous ribonucleotide based on the complementarity. Complementarity means C pairs with the G and adenine pairs with the uracil, not thymine, because thymine is not there in the RNA. So this RNA polymerase will add a ribonucleotide complementary to the template DNA strand. This unwound double helix along with the RNA polymerase core enzyme and growing RNA chain is known as transcription bubble. So transcription bubble is formed during the elongation step. This RNA polymerase enzyme has both unwinding and rewinding activities. RNA polymerase will continuously unwind the double helix ahead the polymerization site. Meanwhile, it will rewind this complementary DNA strand behind the polymerization site. So that is about the elongation step. Third step is termination. Termination of the transcription begins when the RNA polymerase encounters stop signal or termination signal. Stop signal or termination signal is nothing but the sequence of DNA that are rich in GC base and AT base. So it's a sequence of DNA that is rich in GC base followed by AT rich base. When this RNA polymerase encounter this termination site, termination of the transcription begins. There are many proteins involved in the termination process. For example, Rho factor. The newly formed RNA has a binding site for this Rho factor. So this Rho factor will go and bind to the binding site of the RNA, and this Rho factor will move along the length of the RNA until it reaches the termination site. Once it reaches the termination site, this Rho factor will pull this newly formed RNA. As a result, newly formed RNA or nascent RNA will fall off. Simultaneously, the RNA polymerase as well as the Rho factor will be released from the transcription bubble. This results in the termination of a transcription. So by the end of whole process of transcription, RNA is produced or synthesized, and this newly formed RNA in the case of prokaryote is active, or it does not require any processing to become active. So active RNA is produced at the end of transcription in the case of prokaryote. Most of the time in prokaryote, translation also begins before the transcription ends, or simultaneously translation begins before the transcription ends. So that is all about the steps in transcription. I hope this video will be useful. If you like this video, please hit the like button and subscribe to my channel. Thank you.